Hello guys. Today we are talking about the systemic lupus erythematosus treatment.
Start kar, start
Like
kar.
in that, like we are creating an article like about we don't even know about the pathological process, how it started, and according to the pathological process, talked about the treatment methods also, like in our countries, like in our world, like what we are following now, and we're gonna discussing about now, that. And please, guys, introduce yourselves now.
My name is Jaswant Singh. I am from Group 14.
I am Salima Mohammed.
Yeah. Next, please.
Myself Joshua from Group 14.
I am Mohammad Azhar Khan from Group 14.
Yeah, my name is Dhanush, like I'm from Group 14. Let's start with Azarkan. Jason, can you do the screen sharing thing? Azarkan will start the
Yes, yes.
introduction.
Okay, okay, Dhanush.
Okay, Azarkan, you can start now. And can
Okay.
you help with Jashwant scroll that article when he's complete that thing, okay?
Okay. Go down, Jashan.
No.
So my topic is introduction. So systemic lupus erythematosus. It is a virgin autoimmune disease distinguished by the generation of antibody accurate of the immune complexes, exerting effect across the diverse organs. And the origin of SLE is indirect, involving a complex interplay of the genetic, environmental, and hormonal factor. However, the detailed pathogenesis of SLE is remain known, but the central of the indecision and progression of SLE is the breakdown of the self-tolerance. Both innate and adaptive immune response direct at the self-antigens and set in the motion the production of the autoantibodies and deposition of the immune complex with the tissue. The cascade activated the complicated pathway and result in the neutrophil and monocytes come and trigger the activation of the self-reactive lymphocyte. Despite the improvement in the survival rate of the individual with the SLE over recent decade. Further headway in the preceding disease outcome is repeated by the organ damages stemming from the disease itself and the adverse effect linked to the conventional therapeutic approaches. A compression exploration and the intensity of the disease pathogenesis hold significance and the novel therapeutic target and biomarkers specific to the SLE. The review encapsulated and synopsis of the recent advancement in underlying the immunological pathogenesis and targeted associated with the SLE. Okay.
Salima, you will be starting next.
Okay, today I'm talking about dysregulation of the innate immunity. It is so in this manner. So I will discuss about three things: dendritic cells and pattern recognition receptors and neutrophils. In the recent studies, we found that, or the revealing of this, the relationship between the dendritic cells and the type 1 IFN and the patients with SLE. Mostly half of the patients with SLE have the gene expression of IFN alpha in their blood smear with the mononuclear cells. This IFN alpha breaks the autoimmune system tolerance by activating the lymphocytes and dendritic cells and natural killer cells. In case of interferons, it activates the immune system by producing auto antibodies by exogenous process, which is with the type 1 IFN, which forms the B cells for nuclear antigen components of apoptotic cells. And when the rate of apoptosis increases, then it clears the suboptimal region of the system, so that when the rate of apoptosis increases, it also leads and increases in the auto antibody complex, which also considered as a endogenous process for the type 1 IFN. So it promotes the activation of T helper cells, which promotes the presentation of DC cells. which includes the production of the other interleukins like IL-1, IL-2, IL-23, IL-6, etc. So it affects the expression of mitochondrial genes and actin potash channel, so that it causes the disorders of many metabolic disorders. And then the interaction between dendritic cells and PRR complex will be um, talked. And then the dendritic cells is naturally an antigen presenting cells which activate the T cells and by producing auto antibodies. ఇట్లా మాట్లాడితే నేను చెప్పను. Proliferation differentiation of B cells, so that it helps in the crucial role of innate immunity and then activation of adaptive immunity. So apoptotic cells are removed suboptimally and exposed the prolonged period, so that it causes the immune complexes and breaks the immune tolerance again. So coming to the PRR, there are three types of PRRs, which is toll-like receptors and NLRs and RLR. And in this innate immunity, only TLR will plays major role, so that the SLE patients will have more production of anti-nuclear antibodies, which depends on the endosomal TLR and interferon regulatory factors, and signaling pathway produces the type 1 um, IFN, so that dendritic cells help in the endocytosis process, which also maintains and amplifies the immune complex, which continues the ribonucleic proteins and nucleosomes. And in the neutrophil session, we will see the involved action of inflammation and infection, so that when the improper activation of neutrophils happens, it causes the tissue damage in the SLE patients. And then inactivated neutrophils can release the activation of neutrophils can release a large number of cytokines and chemokines, which leads to the immune regulation disorders. And then neutrophil extracellular traps were seen in the patients with SLE, which is a suboptimal clearance of the NET. And excessive NET formation can be involved in the pathogenesis of SLE. Most studies have revealed that NFT formation and defective NFT can cause the autoantigens, which induces the type 1 IFN synthesis and endothelial damage, which also activate TLR. By activating complement classic pathway, it inhibits the degradation of NET, so that the fact of this includes lupus glomerulonephritis is observed in the NET formation, so that there is a decrease in ubiquitination um, in these patients with SLE.
Jashwant, can you scroll down
Thank you.
the article? No. Here, can you scroll down article? Jashwant. Can you something? Yeah. Yeah, here. Now I'm talking about the dysregulation of the adaptive immune system. Like in adaptive immune system, like we're gonna do see like two type of cells, like normally three type of cells. Natural killer cells also was the part of in the adaptive immune system, but it's not like a self-regulated like as like in the T cells and B cells. And these T cells, like in that there are two type of cells, like helper and effector cells. Like in that the helper cells activate the B cells like thing, and in individual affected by the SLE, in individuals affected by the SLE, like alterations will happen in the signaling pathway of immune cells, particularly T cells, influencing the pathogenesis of the disease. And T cells express the surface markers like CD3 in pivotal in the response of the antigens on the context of the SLE in a diminished presence of the signaling CD37 is observed. Like the recruitment of the Fc receptor leading to the heightening of the calcium influx into the T cells and subsequent augmentation of calcium response intensify the immune reaction. Yeah, can you scroll down, Jashwant? Yeah, and there are so many pathways like CD44 signaling pathway and PI protein kinase pathway. This protein kinase pathway is mainly included in so many diseases like hypertension, obesity, and also diabetes. This protein kinase pathway, when it's get activated, it's going to lead to so many chronic systematical problems. And also this CD44 pathway, it's a cell surface molecule integral to T cell activation, exhibits an elevated phosphorylation level in extra extramembrane protein in SLE. This event enhances the T cell addition and migration. The involvement of the kinase ROCK in the process may influence the production of the specific immune molecules. And uh, uh, and yeah, let it be there. Yeah, immune imbalance, like imbalance of the Th1 and Th2 and also Th17 cells. These are the cells like which will be helpful like in the normal body, but like in this imbalance it will leads to the basic cytokine production exhibit the prototype balance in SLE, compromised functionally in Th1 cells, coexist with an overactive Th2 response, resulting in the excessive B cell activation, heightened autoantibody production, consequently tissue damage mainly. As we seen, like it can also lead to the imbalance of the Th17 imbalance. Like Th17, these are the regulatory cells like in our body. These cells will regulate the how much the immune function must be. Like otherwise, if it is deficiency, it will be like that immune actions will not be regulated in our body. That's why it leads to the autoimmune diseases. And T follicular helper cells, like these are the cells like which will present the antigens to the B cells, like like these after this presenting of these B cells, like they will proliferate in the germinal centers of the lymph nodes and they will mature into the plasma cells and they will produce the antibodies like according to the virus, according to the bacteria, according to the which virus they're facing now. And in this, like when we're gonna see the interleukin 21, it is the cytokine which is crucial in regulating the B cell response. And can you go down, Jashwant, some bit? Yeah, no. From here, the Jashwant will continue. Please, Jashwant, you can continue now.
Okay, then. So here, like you can see, T follicular cells, these are cells like play critical roles in the pathogenesis of the SLE. So now I will continue about classic TB cell interactions. These are interactions like when B cells, which are type of the immune cells, encounter specific molecules containing RNA or DNA, they become activated. Or once activated, B cells interact with like CD4 plus T cell receptor. These interactions result of secretion of the various cytokines, like example like TNF, IFN, gamma, IL-6, IL-10. Additionally, CD4 plus T cells transform into the T follicular cells, and which produce IL-21 and IFN gamma. So T follicle cells play a role in the formation of the germinal centers, substanding of the express of the beta cells, lymphoma 6. So now B cell activating factor. It's like protein involved in the interaction between T and B cells interactions. These individuals with systematic lupus erythematosus, these are an overexpression of BAFF, leading to the like increased proliferation of the B cells and survival of the like self-reactive B cell of prolonged. Studies in the mice and lupus models shows that elevated BFF levels are associated with the severe B cell proliferations. So like B cell subsets, regulatory B cells, also called Breaks. Regular B cells, also known as Breaks, have a special role in the regulating in the like immune response. They achieve the by secreting molecules like IL-10 and transforming growth factor uh, beta. Also, IL-10 like interleukin 10 help in inhibit the production of the harmful immune signals, affect the activity of lifespan of B cells, and regulates antigen presentation. Uh, like breaks were found, like regulatory B cells were found in the IL-10 influencing T cell dependent immune responses. In the people with suffering from SLE, the number of breaks is often lower, especially those with lupus affecting the like kidneys. After a treatment of the SLE, they are suppress the immune system. The number of the regulatory B cells uh, tends like to increase. Additionally, regulatory B cells individually like with. In summary, we can see like SLE involves a abnormal response of the B cell leading to production of the harmful antibodies. The interaction like between T and beta cell. Uh, so, Anush,
Okay, Jashwant. Okay, thank you, man.
who will continue?
Yeah, thank you. Joshua will continue about the treatment, like how
Yeah,
we gonna here telling means like here we know the pathology, how it's happening, like the cells, it's the key maintenance of the our immune body. They are causing the like dysregulation in them, like leading to this phenotypic disease. Like for them, we are curing, like according to the by inhibiting them or by blocking them. We have some clinical trial medicines like anafleramab, rituximab, anything. And my friend Joshua
dupilumab.
is gonna explaining that. Joshua, you can continue now.
Yeah, about innate immunity. When we are treating about innate immunity, we have to say that like when we are doing the taking the innate immunity, we have to spotlight it from the interferons and particularly interferon alpha. This can be mainly contributors of the systemic lupus erythematosus, and there is some medicines that we were researched of the this disease, systemic lupus erythematosus. So first one is about anifloramab and rotilimab and cefilenamab and interferon alpha, kenob, and plasmacytoid dendritic cells. When we are talking about anifloramab, it is a fully IgG1K uh, and monoclinical antibody. These are the direct antagonist agonist about type 1 interferon receptors. These are the clinical trials when we are demonstrating the pronounced therapeutic effect in systemic lupus erythematosus. This can be found from the baseline, and investigation are involved in the lupus nephritis and efficiency of the safety profile further. And rotonilimab, these are the anifrolumab, and these are the anti-interferon alpha agonists evaluated in the systemic lupus erythematosus. These are the most efficiency that can be formed from the individuals of low interferon signatures. When we are treating from the meeting primary endpoints, the cefilimab unfortunately demonstrate no efficiency. That did not progress advance the development, and the new interferon monoclinical antibodies are formed, such as AGS-009 and JNJ-559-20839 and AMG-811. These are the currently undergoes assessment in the Office of Clinical One trials. And when we are talking about interferon cninoid, these are the vaccine comparing from the interferon 2B coupled to carrier. These are the mainly proteins that present in the interferon alpha blockers, and these are mainly targeting the antibodies of INFA subtype. These are the significantly expressed in the interferon signature that can be formed in the placebo and differences in the diseases. And these are the activate the score of a serum C3 and C4 antibody, anti-dsDNA. These are the concentrated were not observed. When we're talking about plasmacyte dendritic cells, these dendritic cells mainly produce from the capability of interferon alpha, targeting from the plasmodic dendritic cells. These are directly act on the long antiseptic therapeutics in the systemic lupus erythematosus and encompassing the anti-blood dendritic antigen two bodies, anti-CD123 monoclinical antibodies and BCL2 inhibitors undergo evaluation of the distinct phases of the clinical trials and modulating the interferon dynamics in systemic lupus erythematosus pathogenesis. When we are talking about adaptive immunity, there are mainly three to four types of the this affect the T cell of the target therapeutics. First one is abatacept and CD40 CD40L interaction. These are the T cell targets, and B cell target therapies are belimumab and rituximab, exploring of the surface antigens. And T cell targeting therapies are abatacept. These are the drug mechanism that can be attached to the part of the IgG, and these are the extracellular domain of the CTLA4 fusion that can be higher efficiency for CD80 or CD86. By blocking the T and B lymphocyte co-stimulation, Adacept inhibit the T cell activation and preventing the B cell responses. These are mainly affecting from the arthritis. And deficiency of systemic lupus erythematosus remain the mixed results in clinical trials. When we are talking about CD40, gives rise to CD40L. These are the receptor of the ligand pairs are differentiated by differentiation. These are the cell differentiation. BG9588 and anti-CD40L monoclinical antibodies faced the early discontinuous thromboembolic events. Another anti-CD40L monoclinical antibody, these are the dapirolizumab, is currently under the assessment. There are various agents targeting the receptor and ligand pair are explored. But talking about B cell therapies, the drug is belimumab. These are the human IG1 gamma monoclinical antibody inhibit the B cell survival differentiation by blocking the soluble BLYS. These are the approved treatment for the active autoantibody positive SLE, demonstrated by superior therapeutic response in the subpopulation. These are the higher baseline diseases activity. A proliferation induced ligand or the similar BAF influenced B cell survival activation. These are the responsible for the Accept target APRIL. Other B cell targeted agents are under the evaluation. Rituximab, these are the human mouse chimeric monoclinical antibody against CD20 receptors. These are the induced B cell apoptosis, induced proliferation and effective addresses. These are the abnormal B cell proliferation approved by treating B cell lymphomas. And clinical benefits about SLE manifestation include nephritis and neuropsychiatric diseases, adverse event like allergic responses, anti-CD20 antibodies such as ofiftimab and optinozumab. These are the investigated. Other than these drugs, there are some surface antigens. These are the therapeutic targets of CD22, CD19, and CD74 are explored due to expression of the across differentiation B cell subsets or approach aim to modulate B cell responses and apoptosis, deletion and energy addressing the loss of B cell tolerance observed in a SLE. And these are the production of antibody against nuclear agents. This research in the area aims to enhance the efficiency of B cell targeted therapies in the treating the systemic lupus erythematosus. Thank you.
So in the conclusion, we can see SL is a multi-fractal like autoimmune disease that can affect nearly every organ in the body. In conclusion, we can see like in the past 50 years, this disease have moved from the chronic steroids and high dose of chemotherapy like regimen to target biological therapy. SLE stand as like a complex autoimmune disease characterized by the multitude of cellular of molecular abnormalities making it a pathogenesis of subject of ongoing investigation. Despite like significant steroid compressive understanding of SLE pathogenesis remains elusive. The not self tolerance immune response in like systematic lupus erythematosus is that extensively covering both innate and adaptive immunity. Advance in our compression of cellular and molecular phenotypes in SLE pathogenesis have expected significantly. Like we found B lymphocytes emerge central place in SLE adaptive immune response, contributing of auto antibody production, autoantigen presentation, and autoreactive T cell activation. T lymphocytes, meanwhile, they are influenced through co-stimulation mediated signaling pathway and cytokine secretion of the bispecific T cell subsets. Innate immune response also play a critical role in the SLE pathogenesis, particularly the discovery of the toll-like receptors, TLR receptors, and also plasmacytoid dendritic cell. These receptors can be activated by immune complexes, triggering the production of the interferon alpha and yet like formation of neutrophil extracellular traps. These are abnormalities complement each other. These
Thank
are
you,
like
Jeshwant.
references.
Okay.
Uh,
Yeah, thank you, Jeshwant. Thank you guys for the meeting. You can leave now.
Thank you.